Wait, did I take a picture?
No, it's recording.
Yeah, you're right.
Three.
On three, are we ready?
You're good. You're good.
We're good to go. You can see the ovary right here. And what they're representing in the ovary, these primordial follicles, because they only have maybe 1, 2 layers of cells at best. Then when they have multiple layers, 2 to 3, 3, 4, it becomes primary follicle. You can see this is also a primary follicle. It will become secondary when it not only has multiple layers of cells, but it starts developing this thing called an antrum. So I would call this a secondary follicle. The antrum will increase in size, such as this. See how it's increasing, and the follicle gets closer to the periphery of the ovary. This is a mature or Graafian follicle. You can see the oocyte here with some cells surrounding it here and here. You can see ovulation took place. Some cells were left behind. They will form the corpus luteum, and if you don't have implantation occur, it regresses to a corpus albicans. The ovulated egg have cells around it, which is the corona radiata. The white ring is the zona pellucida, and then you have the oocyte right here, which I believe it's called secondary oocyte, or secondary oocyte. You have the fimbriae right here, fimbriae of the fallopian tubes or uterine tubes. The region where they're attached to is called the infundibulum. When it starts to curve, this is called the ampule of the uterine tube. You notice that you have the egg in here that's been ovulated and sperm going to fertilize it. If it does fertilize it, it will undergo mitosis, 2 cells to 4 cells, 4 to 8 to 16 to 32, 64, and so on, until you go down the uterine tube and implant in that endometrial lining. Up here is you'll see a representation of the egg and sperm only. The purple on here, we've taken away the corona radiata and the zona pellucida, so this indentation would represent those cells in that layer. You can see on here the corona radiata, which is a cell, the zona pellucida, and the oocyte here. You can see the 4-cell stage here. You can see possibly, 64 or 128 here. This would be referred to as a blastocyst. It will develop a hole in it called the blastocele. Some of the cells on one end would become part of the inner mass. The other cells on the periphery would become part of the trophoblast. When you start developing the cells in the inner mass on one end, they differentiate. And here's a cross-section of that differentiation. You'll notice some of the cells here will differentiate into one structure and another one down here. This is that bigerminal layer that we talked about in lab. One end of it will become the yolk sac, and the other end of it will become part of the embryo and other embryonic structures. You can see in this cross-section right here, you have the yolk, you have the conceptus or developing embryo. It is a neural tube right here. The ventricles would be here, the vertebrae would be here. You can see on the outside, the amnion is being developed. And I'm assuming these are part of those cells, the corona radiata, that are surrounding the entire structure. You can see right here, in terms of the embryo developing, you have the individual primary brain vesicles. You can see these 3. You can see the optic cup with the actual lens blocode. So the optic cup is everything, with the lens blocode, maybe the bluish structure here. At the very least, you have the heart here. You have the liver here. You have your limbs, such as arm here, legs here, the vertebrae, which is here, the umbilical cord right here. And that's it.